Welcome everyone. In this video, I am going to discuss about the third part of cell cycle, which includes regulated synthesis of CDKs and cyclins, cell cycle checkpoints, role of retinoblastoma, and p53 protein in cell cycle. In regulated synthesis of CDKs and cyclin, I forgot to mention one mechanism in previous video. So in this video, I'm going to explain that one. In this mechanism, the changing CDK activity is the regulation of the rate of synthesis of cyclin and CDK or both. Cyclin D, CDKE, CDK2 and CDK4 are synthesized only when a specific transcription factor E2F is present in the nucleus to activate transcription of other genes. Synthesis of E2F is in turn regulated by extracellular signals such as growth factors and cytokines. This is the compounds found to be essential for the division of mammalian cells in culture. These growth factors induce the synthesis of specific nuclear transcription factors essential to the production of the enzyme of DNA synthesis. Growth factors trigger phosphorylation of the nuclear protein Jun and Fos transcription factor that promote the synthesis of a variety of gene products including cyclin, CDKs and E2F. In turn, E2F controls production of several enzymes essential for the synthesis of deoxynucleotides and DNA that enabling cells to enter the S phase. That means growth factors or cytokines gives the signal in the production of E2F, cyclins and CDK through the phosphorylation of Jun and Fos nuclear proteins. This is the diagram which explains the previous slide, where the growth factors and cytokines phosphorylate the Jun and Fos proteins present in nucleus. This phosphorylation occurs through the MAPK cascade. MAPK means mitogen activated protein kinase. These mitogens are the signals that act from outside of the cell to inside mitosis and cell division. The phosphorylation of Jun and Fos proteins in nucleus produce cyclins, CDK and transcription factor E2F. This transcription factor synthesize the enzymes required for DNA synthesis, and that enzymes will take part in the S phase in the DNA replication. Okay. From that we finish that regulation part, and next is cell cycle checkpoint. The cell cycle is a highly regulated and a series of events that is mediated by a number of checkpoints. As you have seen my previous video, there are three checkpoints are there: G1H checkpoint, G2M checkpoint and M phase checkpoint. These checkpoints functions to ensure that incomplete or damaged are not replicated and passed onto daughter cells. Okay. In most cells, there are several checkpoints in the cell cycles at which the cycle can be arrested if previous events have not been completed. In G1 phase, cell integrates mitogenic and growth inhibitor signals that makes the decision of the cell to proceed, pause or exit in the cell cycle. In case of yeast and mammalian cells, the G1 is the most important checkpoint where the cell becomes committed to DNA replication and completes the cell cycle. Two fundamental checkpoints, replication checkpoints and spindle checkpoints, operate in every round of the cell cycle. Okay, replication checkpoint is present in G2M and the spindle checkpoint is present in M phase. The replication checkpoint monitors progress of DNA replication and arrests cell cycle at G2M transition until DNA replication is complete. Whereas spindle checkpoint monitors attachment of chromosome to mitotic spindle. They prevent segregation of sister chromatids until they are properly aligned on the metaphase plate. Okay. Perturbation means cellular illness in the cellular environment can activate additional checkpoints that leads to observed checkpoints and inhibition of cell cycle progression. In a cell, when DNA is damaged, it needs to be repaired. So, the cell activates DNA damage checkpoint to arrest cell cycle. According to cell cycle stages, DNA damage checkpoints are classified into at least 3 checkpoints. First one is G1S checkpoint, second one is intra-S checkpoint, third one G2M checkpoint. The G1S checkpoint inhibits the transition in cells that have not yet committed to DNA replication. Intra-S phase checkpoint prevents initiation of DNA replication at origins that have not yet been activated. G2M checkpoint inhibits entry into mitosis. Okay. During mitosis, chromosome segregation is carefully monitored to prevent aneuploidy. I will tell about aneuploidy in my next video. Okay. The spindle assembly checkpoint and mitotic checkpoint. Guarantees that anaphase onset is initiated only when all chromosomes are properly attached to microtubules and aligned at the metaphase plate. The spindle checkpoint blocks anaphase entry by inhibiting the anaphase promoting complex. When kinetochores do not receive spindle fiber from both the pole of the cell, protein MAD2 binds to CDC20 and does not allow the APC to bind to CDC20. Okay. APC is the ligase enzyme. It will not be attached to CDC20. At the same time, that MAD2 is sequestering CDC20. The centromere protein E activates BUBR1, which also block anaphase. Okay. So when kinetochore does not receive spindle fiber from both the pole, MAD2 will attach to CDC20, and as well as this MAD2 also sequestering the CDC20 and activates sequestering CDC20 and the centromere protein E presence in the cell activates BUBR1, which also block anaphase. Okay. The localization of MAD2 and BUBR1 to kinetochore may be dependent on the Aurora B kinase. Cells lacking Aurora B fails to arrest in metaphase even when chromosomes lack microtubule attachment. Then we will see the role of retinoblastoma protein in cell cycle regulation. It is a protein of Rb gene. Rb gene means retinoblastoma gene. That is also called tumor suppressor gene. Then question is coming that how the product of Rb gene regulate cell division. Rb is a substrate for CDK Cyclin D complex and exert its effect during the part of G1 that precedes the restriction point. It acts as a brake in mammalian G1 cells. The transcription factor E2F binds to specific DNA sequence and induce many genes to transcribe. High level of E2F activates transcription of Cyclin A gene. The product of this gene is required for S phase of this cycle. But when Rb protein binds to E2F, the activity is suppressed and transcription of gene involved in S phase stopped. Okay. The phosphorylation of Rb protein reduces its affinity for E2F and allows E2F to heterodimerize with factor Dp. Dp means DNA binding proteins and activates the S phase gene expression. Phosphorylation of Rb protein is initiated by Cyclin D CDK4 and Cyclin D CDK6 in mid G1. On or hypophosphorylated retinoblastoma protein blocks cell cycle progression. In homozygous null Rb cells, Rb protein is permanently inactive. Thus, E2F is always able to promote S phase, and the arrest of normal cell in late G1 does not occur in retinoblastoma cell. So we have understood that generally when retinoblastoma is present in with E2F, it is present in inactive form. The complex is inactive. But when Cyclin D CDK4 and Cyclin D CDK6 is coming, they phosphorylate the E2F, they phosphorylate the Rb protein and make the E2F active. That act as a transcription factor for genes whose product are required for entry into S phase. Okay. Next we will see the retinoblastoma. Retinoblastoma is a childhood tumor of the retina. The gene whose loss is critical for development of the cancer is called retinoblastoma or Rb gene. The retinoblastoma arises when both copies of the Rb gene is inactivated. There are two forms of the disease. One is hereditary and other is sporadic. The hereditary form individuals inherit a single defective copy of Rb gene, and tumors occur where the remaining normal copy is lost or inactivated by somatic mutation. They develop retinal tumors early in life and generally both eyes. In the non-hereditary form or sporadic form, all cells initially contains two functional copies of the Rb gene, and the tumor arises because both copies are lost or inactivated through the coincidence of two somatic mutations in one cell. Because this is an unlikely occurrence, sporadic retinoblastoma is rare, develops late in life, and usually affects only one eye. So the sporadic form affects only one eye and hereditary form affects in both eyes. Okay, so in hereditary form it inherit only one copies and other copies are inactivated by a somatic mutation. Here inherit two functional copies are lost or inactivated through the two somatic mutations in the cell. So both are inactivated. Both copies are inactivated. Then we'll see the role of p53 in cell cycle regulation. DNA damage blocks progression through the cell cycle until the damage is repaired. Damage to DNA can result from chemical and physical agents. Tumor suppressor protein p53 contributes to arrest of cells with damaged DNA at DNA damage checkpoint. Cells with functional p53 arrest in G1 and G2, whereas cells lacking functional p53 do not arrest in G1. This p53 protein is a transcription factor, regulates the expression of cell cycle regulatory protein. p53 is not required for normal cell division. Thus, under normal conditions, it is extremely unstable. The proteasomal degradation of p53 results from its polyubiquitination by ubiquitin ligase MDM2. In case of DNA damage, DNA-dependent protein kinase ATM and ATR are activated. ATM primarily is a sensor of DNA defects caused by ionizing radiations, while ATR is more specialized for UV-induced DNA damage and inhibitors of DNA replication. In case of DNA damage, the rapid degradation of p53 is inhibited by ATM, which phosphorylates p53 at a site that interferes with binding to MDM2. Hence, in response to DNA damage, p53 levels increase and arrest the cell at the G1 phase of the cell cycle. Okay. If all of the repairs have been made to the DNA, the cell divides normally and completes the cell cycle. However, if the cell still contains mutated or duplicate DNA sequence, it dies by suicidal apoptotic mechanisms to prevent its proliferation. In those cells that have mutated or lost p53 gene, the arrest at G1 does not occur, and the cell that have mutated genome proliferate and become cancerous. This is the diagram how the DNA damage stopped the S-phase CDK through p53 proteins. So when DNA is damaged, it triggers to activate ATM kinase enzyme, and that kinase enzyme phosphorylate the p53 protein generally presents in combination with MDM2. MDM2 is ligase enzyme, and when it is attached to p53, it is in inactive stage. So this ATM kinase phosphorylate the p53 and make it active by separating this MDM2 and p53. When p53 is active now, it acts as a transcription factor for the synthesis of the p21 protein. So when p21 is synthesized, it binds with S-phase CDK, and make it inactive. So the process is stopped. So if we will see the overall figure, so when DNA damage is there, they activate the p53 protein, and p53 will activate p21, as you have seen in previous video, the previous slide. The p21, when p21 is activated, synthesized, it attached to the CDK, cyclic CDK complex, activated cyclic CDK complex, and make it inactive. Okay. And progress of cell cycle will stop. Generally, what happens when Cyclin CDK complex is activated, it also phosphorylate the retinoblastoma protein, because generally retinoblastoma proteins are in inactive form when it will be combined with E2F. Okay, so active cyclic CDK complex phosphorylate the retinoblastoma protein. So this E2F transcription factor that is produced by the Jun and Fos protein, the Cyclin CDK and E2F is produced as you have seen in previous slides. that is that Jun and Fos is phosphorylated through MAPK cascade, and that cascade that get the signal through growth factors and cytokines. And this E2F, one way it will be attached to p53, that is the retinoblastoma protein, and another way it will transcribe the enzymes, synthesize the enzymes required for DNA synthesis, and that will pass from G1 to S phase of this cell cycle. This is the actual role of p53 in the cell cycle, and that's all for this video. We will discuss on another video on the other topic. That's all. Thank you.